Today topic of discussion PAP staining. Uh, it is a staining method routinely used in cytopathology laboratory. This technique, after the name of scientist Dr. George N. Papanicolaou, the father of exfoliative cytology. It is a polychromatic staining method. Poly means many, chromatic color, many coloring staining method. This PAP staining mainly used for deduction of both gynecological and non-gynecological example, that female genital tract, non-gynecological sputum, CSF, like that. So vaginal and cervical, then body fluid like cerebrospinal fluid, peritoneal fluid, other specimen like sputum, urine deduction we are using. This one is the reliable technique in cervical cancer screening in gynecology. So it is a reliable technique in cervical cancer screening. It is a polychromatic staining containing multiple dyes to differentially stain various component of that cell. So it will do staining various component of that cell. Next one is that principle of PAP staining. PAP staining containing both acetic dye and basic dye. Acetic dye stain containing basic component of the cell. Basic dye stain containing acetic component of the cell. So what are the dye here? Example, hematoxylin. It is a basic dye stained for nucleus. So what is the role? Basic dye stain acetic component. So nucleus is the acetic component. Then eosin, it is a acetic dye. It is stained for cytoplasm. So cytoplasm is the basic component. So here it having both acetic and basic dye. Acetic dye stained for basic component. So eosin is the acetic dye. This one staining for cytoplasm. That one is the basic component of the cell. Then basic dye, hematoxylin. It is a basic dye. This one stained for acetic component of cell. Next one is requirement, multiple coupling jar, then polychromatic PAP stain, including here 5 dyes in 3 solution. So here we are using 5 dyes in 3 solution. Alcohol, then distilled water, 5 dyes. What are the 5 dyes? First one is hematoxylin. Then this hematoxylin, here mainly using Harris alum hematoxylin. It is the neutral dye used for nuclear staining. This Harris alum hematoxylin, it is a natural dye used for nuclear stain. It containing the component hematoxylin 1 gram, then alcohol 10 ml, ammonium or potassium alum 20 gram, distilled water 200 ml, then mercuric acid 0.5 gram. Second, that dye orange G OG6. That means orange green 6. This one acts as a first counter stain. First counter stain. Here this OG6 used for staining to cytoplasm. This one is the first acidic counter stain. Then it will do staining in squamous cell carcinoma. Then stain matured and keratinized cells. Then we know totally 3 dyes in 3 solution. Already 2 dyes we discussed. Now remaining 3 dyes. Third one is 3 dyes together. We have that name. This eosine assure. Eosine assure, it having that component EA36 and 50. This one is the second counter stain. So third dye, 3 things together using Y. It gives pink color the cytoplasm of mature squamous superficial epithelial cell RBC and cilia. That eosine Y, it totally 3 dyes. First one is eosine Y, gives pink color the cytoplasm of mature squamous superficial epithelial cells RBC and cilia. Then light green SF. This dye yellowish stain the cytoplasm mast cell paraphasal cell. Then Bismarck brown Y stain mucin. So these 3 dyes together we have that name using azure. So first counter stain orange G6 and EA36 for second counter stain. Next one is procedures. That fixative is used for PAP smear is ethanol 95% of alcohol or 95% of alcohol. So the fixative is used for PAP smear is ethanol 95%. The following solutions are required for PAP staining: descending grade of alcohol for rehydration. Descending grade of alcohol for rehydration. Take the slide from 95% of alcohol transferred into 70% of alcohol and give 10 tips. Place into 50% of alcohol. 10 tips. Each 95%, 70%, 50%. We will give 10 tips. 10 times we want to do tipping. Here the descending series of alcohol 95, 70, 50 used to rehydrate the cell. Rehydration help the water to get into the cell component. So here rehydration main purpose the water to get into the cell component. After descending series of alcohol, each and every step 10 times we want to do tipping. That 95%, 70% and 50%. After that we want to wash gently in distilled water for 15 seconds. After that we want to do staining. That differentiation is the here differentiation process is the process that involve the removal of excess stain until the color retain only in that appropriate cell component. So here staining we are using Harris hematoxylin for 2 to 3 minutes. Here staining we are using Harris hematoxylin for 2 to 3 minutes. After that we want to wash with water. Then tip 0.25% of HCl 2 to 3 times. First staining. Here differentiation process we want to do. This method remove excess stain until the color retain only in that appropriate cell component. Then rinse that distilled water. Then tip in 0.25% of HCl for 2 to 3 times. then place under running tap water for 50 seconds. Then dehydration. Dehydration process removal of water. Since alcohol can be penetrate the cell quickly and water can be easily replaced with alcohol. So the dehydration process removal of water, since alcohol can be penetrate the cell quickly and water can be easily replaced with alcohol. Rehydration through ethyl alcohol. Here we want to follow ascending order 50, 70 and 90 percentage. 6 time each solution, 6 time we want to do tipping. Then stain in orange green OG6 for 2 minutes. Then rinse in 95% of alcohol for 10 tips. Then we'll do stain in using azure 50 and 36 for 2 minutes. Then dehydrate in absolute alcohol 95 and 100%. After that, clearing process, clear in alcohol xylene. It will do 3 changes. After that, mounting TPX mount in neutral synthetic medium after drying on the smear. Then result, nuclei we can observe blue with clear sharp detail. Then cytoplasm wearing shade pink, blue, green, yellow color. Cytoplasm shade become vary pink or blue or green or yellow, but nuclei blue with clear sharp detail. So this one is the summary. First one is the fixative is used for PAP smear 95% of alcohol 1 to 2 minutes. Then the rehydration with the help of water get into the cell because of rehydration process. Then tip 5 to 10 times in 95%, 70%, 50% alcohol. Then hematoxylene 2 to 3 minutes. Then tap water for 15 seconds. Then 0.25% of HCl 2 to 5 minutes. Then tap water for 15 seconds. Then ethyl alcohol, here dehydration process 50, 70 and 95%. Then using OG6 2 minutes. Then alcohol 95%, 10 tips. Then using azure 36 and 50 2 to 3 minutes. Then absolute alcohol. Then xylene, it will causes 3 changes, 5 tips. Then TPX mount with cover slip. We want to observe result in microscopy. This mounting is the process in which used to keep the stained slides for longer period. Okay, by using that mounting, keep the stained slide for long period and label the slide and observe under microscope. Mounting the process with it's used to keep the stained slide for long period and labeled the slide. After that we can observe under microscope that nuclei blue color, cytoplasm pink or blue or green in color. So this is all about PAP staining. Thanks for watching.